Hi everyone, I'm Kvitne. I'm a postdoc at UC San Diego, and the title of my talk today is Distinct Fecal Microbial and Metabolic Signatures in Very Early Onset IBD. Since all of you might not be familiar with what very early onset IBD is, I want to start by just giving a brief overview of the disease. This is a form of IBD manifesting in children less than 6 years old. Compared to IBD diagnosed at later ages, it's often associated with a more severe disease course. The clinical presentation often varies a lot, which means that there is often a delayed time to accurate diagnosis, and patients overall have a lower response to conventional treatment. And the incidence of VEO-IBD is rapidly increasing worldwide, meaning that we need to do better to understand its multifactorial etiology to improve outcomes for these children. The gut microbiome is increasingly recognized for its important role in the pathophysiology of both adult and pediatric IBD, but insights into VEO-IBD remains limited. So in this study, we wanted to investigate changes in the fecal microbiome and metabolome in very early onset IBD. So we recruited 26 patients diagnosed with VEO-IBD and 28 sex and age-matched controls. And we then collected one fecal sample from each participant. And the fecal metabolome and microbiome were profiled using untargeted metabolomics and 16S gene sequencing. And we then process data using our standard pipeline, which include Cheetah for microbiome analysis and MCMine and GMPS for the metabolomics, before we did downstream analysis. Here you can see a brief overview of the study population. So mean age was around 6 years in both groups, with an almost 50-50 ratio male and female. Most patients with VEO-IBD received medications either with a TNF-alpha inhibitor or an aminosalicylate, or a combination of these. So let's start by having a look at the metabolomics data. So we first did an unsupervised principal component analysis of RCLR transformed data. As we can see from the figure, there was a clear separation in the fecal metabolome between VEO-IBD and control. And from the Permanova, we can see that both cohort and age had a statistically significant effect. And next, we did a supervised PLSDA in order for us to identify the features that are driving separation between the two groups. And features with a VIP score more than 1 were considered relevant. We then plotted the natural log ratio of these features, and we can see that they are clearly separating the two groups. One of the major classes of metabolites that separated VEO-IBD from control was the N-acyl lipids. N-acyl lipids are molecules that consist of a fatty acid and an amino-containing group linked by an amide bond. And interestingly, we found that actually several of these short-chain N-acyl lipids were significantly depleted in VEO-IBD. And we don't know exactly the role of these metabolites yet, but there are studies indicating that they are important for immune regulation. We also observed a higher relative abundance of both di and tripeptides in VEO-IBD. And this is interesting because a recent study just reported that there is a correlation between increased dipeptide abundance and disease activity in patients with ulcerative colitis. And in this study, the elevated dipeptide levels was linked to an overabundance of microbial proteases produced by the Bacteroides. And consistent with this finding, we also observed a higher abundance of Bacteroides in VEO-IBD. What we don't know yet is whether these peptides directly contribute to the pathophysiology or if they are just byproducts of the altered gut microbiome composition. We also explored the presence of these peptides in other publicly available metabolomics IBD datasets by searching them against TissueMast. TissueMast is a recently developed tool in our lab. It allows you to search MSMS spectra across publicly available metabolomics datasets with metadata on sample type and disease status. So for this analysis, we focused on human stool samples. And if we look at the figure, we can see that all the 5 peptides that we searched were detected more frequently in fecal samples from individuals with IBD than in healthy individuals. And this suggests the possibility that actually maybe certain diatripeptides can serve as biomarkers for disease activity or severity in IBD. We were also interested in the bile acids. The bile acids are cholesterol-derived signaling molecules synthesized in the liver, and once in the gut, they do undergo microbial modification. So when we looked at the discriminant bile acids from the PLSDA model, we saw a clear shift in the bile acid pool between VEO-IBD and control. And this was mainly driven by a higher abundance of oxobile acids, which are bile acids with one or more ketone groups, but also amino acid conjugated bile acids. So moving over to the microbiome, we did not find any difference in alpha diversity between the VEO-IBD and the healthy, which was a bit surprising. However, when we are taking age into account, we do see a moderate positive correlation between age and the Shannon diversity index for the VEO-IBD group, but not for the control group. And we do think that maybe a prolonged exposure to chronic intestinal inflammation and also maybe the medications they're using are delaying microbial diversification. So for beta diversity, which measures a difference in microbial community composition, we did observe a strong separation between the groups using PCA of RCLR transform data. And Permanova confirmed that this difference was statistically significant. Next, we also did a differential abundance analysis using ALDEX to identify discriminant taxa at the genus level. So the output is showing us that there is a higher abundance of taxa such as Lactospira, Veillonella, and Sotorella in the very early onset IBD. And several of these has also been associated with IBD in previous studies. And we also observe a lower abundance of the gut commensals such as Coprococcus and Collinsella. And we also did a PLSDA, which gave us very similar results. As we can see from the figure, we also observed a lower abundance of Bifidobacterium and Blautia in the VEO-IBD, which are taxa associated with gut health. So finally, we wanted to investigate if any of the metabolic changes we detect could be explained by changes in the composition of the gut microbiome. So we did a multi-omics analysis using Diablo. So several of the short-chain N-acyl lipids, such as those with methionine or phenylalanine as head groups, were positively correlated with toxa enriched in the healthy, and then they were negatively correlated with toxa enriched in the VEO-IBD. So as a take-home message, VEO-IBD is associated with distinct alterations in the fecal metabolome and microbiome. These children has a lower abundance of bacteria linked to gut health, such as Bifidobacterium. They also have altered levels of metabolites belonging to the classes of N-acyl lipids, diantripeptides, and bile acids. Overall, these findings do provide us with a foundation for developing targeted diagnostics and therapeutics. For example, manipulation of the gut microbiota composition or function through either pre or probiotics or even fecal microbiota transplantation may provide an opportunity to target processes that contribute to disease in these children. And if you want to read more, feel free to check out my preprint. And with that, I would like to thank all my co-authors, and I would like to thank you for listening, and I'm happy to take any questions later. Thanks.